Good morning, good evening, ladies and gentlemen. My name is Gabriel Miranda. I'm a doctoral student at the University of Brasilia, and I'm going to present today this work that is part of a bigger work that is being done here at the University of Brasilia in Brazil. And it's entitled The Determination of Colorimetric Changes in Blueberry Cultivar Biloxi under Different Doses of Nitrogen Application. Blueberry, it's a crop that has significant increase in agriculture importance in the last years because it's a crop that is very profitable, and also it's a crop that has increased the cultivated area in the world, especially here in South America. And also it's a fruit that has increased the consumption because there are many health benefits, and there are also many good qualities because it's low in calories, it's good for health, for active to work in our body, for combat many diseases. And so the cultivation of blueberry has increased, and this is a good thing. But here in Brazil, the cultivation is very limited to the states of the southern region. That's because of the climate requirements of the cultivars that we have here, that we use here. Most of those cultivars are from the group Rabbit, so are cultivars that demand a high hours of cold to flower and to produce. So that's the reason, because it's very limited to the southern area of the country. But however, recent introduction of those new cultivars that have a 0 to low chilling requirements, it's making it possible to grow this production in other areas of Brazil, and especially here in Brasilia, that's the center of the country and has a tropical climate. So when we talk about blueberries, it's an important factor for us is the color of the fruit. Blueberry is a fruit that has color in its name, so it's already associated with a color. So when the consumer goes to the market to buy the fruit, and he's going to buy a blueberry, he will see the color, and if the color is not good, he will never have the opportunity to try the other attributes of the fruit, as the flavor and the texture. So we focus very much on the flavor and the texture of the fruit, but the color is also an important factor for us to evaluate, because if the color is not good, the consumer will never buy the fruit. And studies have shown that there is a preference for a brighter and less intensively blue-colored fruit. And also the color is associated with the anthocyanin content of those fruits. And also the anthocyanin content is responsible for the blue color in the blueberries. And then the nitrogen is the most important nutrient to maintain the vegetative growth of blueberries. And also it has special functions in other organic compounds in the plants. But the effect of the nitrogen fertilization on the color of the fruits and the color of the epidermis is not yet fully established. And I say that because in different cultures, there are different results of interaction between the nitrogen doses, and also results that show no interaction between the nitrogen doses and the color. So the information of the nitrogen effect on the color of the skin of blueberries is still very scarce, and especially here in tropical areas. And so the objective of this study was to evaluate the colorimetric attributes of southern high bush blueberry, the cultivar Biloxi, according to the different doses of nitrogen in fertigation. So the research was conducted in the pomology sector of the University of Brasilia. This photo is an aerial image of our research station facilities. And the period of study was from August 2018 to July 2019. So the climate of this region is, according to Copenhagen, it's AW. It's we have a dry winter, a very dry winter, and a rainy summer. And the average temperature here ranged from 23.4 degrees Celsius to 19.1 degrees Celsius. And a very important thing to also that I wanted to highlight is that the lowest temperature registered was 8.5 degrees Celsius. And this is important to highlight because this cultivar, we found in the literature that it requires 200 hours of cold to achieve, to flower and to bear fruit. And here in our region, we could have good yield of fruits without any temperature below 7.2 degrees. And this is a very important thing to point out. So the cultivar is Biloxi, and we planted in this system in containers of 60 liters. We used rice husk as the substrate, 100% rice husk, and we used a drip irrigation system that fertigated during the entire cycle. So the experimental design that we used was a randomized complete block design. We had 4 treatments with 10, 20, 30, and 40 grams of nitrogen per plant, with 8 replicates and 5 plants per experimental plot. For the colorimetric assessments, fully ripe fruits were collected from each plant, and we made some samples of 200 grams, and we tested the colors in different areas of the fruits in a spectrophotometer, the ColorQuest XE. And we evaluated the variables according to the C-Lab coordinates, and we got hue angle and chroma as well. And all the data were subject to analysis of variance by the f-tests with a 0.05 significance level. So these equations here are the equations that we used to evaluate and to get to the hue angle and to the chroma value according to the coordinates that we obtained, the L, the A, and B. So the colorimetric reading was performed by these C-Lab coordinates. And just to point out, sometimes this can be very difficult to understand, even for me. And the hue represents the quality of the color. So hue varies from 0 to 360 degrees. And so it represents the colors such as blue in blueberries. So we have the different quadrants of the circle, and the angle will give us in which color our measurement was obtained. So value or lightness is the lightness of a color. It's a darkness of a color, if it's a darker color or a lighter color. So it depends if it represents how the color goes to black or to white. And chroma represents how strong or how intense is the color. So the results that we obtain is that there was a significant statistical difference for the variables in this experiment, for the lightness, for the hue degree, and for chroma. And also we found out that there was a positive effect on the doses of, not a positive effect, I'm sorry, there was an effect on the doses of nitrogen under the parameters of lightness, of hue, degree, and chroma. And this is the graph that we can see with the results that we obtained. And we also saw that the higher value for lightness was achieved under the estimate dose of 24.37 grams of nitrogen per plant. So doses above this dose represented a reduction of the lightness on the fruit skin. Also, doses above 20 grams of nitrogen per plant also caused a reduction on the hue degree. So a reduction on the hue degree represents a blue, represents that the color is going to the quadrant of blue, and maybe if we use more nitrogen, we would have a less blue or violet intense color. So the hue degree varied from 266 to 264 degrees. And it's important to highlight that the hue degree around 240 and 270, it represents a fruit with a blue and violet colors. So it also was observed that for the higher doses of N applied, lower results for chroma were obtained. So the higher chroma was reached with the application of an estimated dose of 15.74 grams of nitrogen per plant. So as a conclusion of this work, it's important to highlight that we found that higher doses of nitrogen reduced the values of lightness, of hue degree, and chroma on the skin of fresh blueberries. And also the skin color that we obtain on all the doses of nitrogen that we use were compatible with the preference of the consumer of color. So all the variables that we use for nitrogen doses, we had a blue or violet color of blueberry. So all the fruits in this plot were good in the color attributes. And this is the results that we have. And I would like to thank the Congress organizers for giving me this opportunity to present this work. And I also would like to acknowledge my team, my supervisor, and all the other grad students and interns that we have in my university. And I would like to thank them all for the support, and also thank the financial support from my university and CAPES, and also FAPDF, because it has given our financial support to perform all this work that we have been done. And feel free to send me your questions, and we can discuss anything in this work later, and I'll be more than happy to answer all your questions. Thank you.